This is part 3 of the viruses lecture. This was the slide that we had gone over in the previous recording. And remember, the central dogma of life, this shows you how transcription and translation occur, how DNA codes for a protein. So how pieces of DNA called genes are transcribed into messenger RNA, and messenger RNA is used to synthesize a protein that occurs on ribosomes, if you remember. So we're going to compare each virus, each of the 7 different classes of viruses, to living organisms, transcription and translation. So group 1 is really easy. The nucleic acid core is double-stranded DNA, and their DNA undergoes transcription and translation inside the host cell. The only real difference is that the viruses use many of the host cell's enzymes. So in a cell that has double-stranded DNA and it undergoes transcription and translation, it uses lots of enzymes like helicase, polymerase, ligase. All these different enzymes are used to carry out transcription and translation and to produce a protein. But the virus is going to have to make use of those enzymes within the host cell. So here's the example, our double-stranded DNA. I'm using the same example each time that I used in the last slide because I think it'll keep things simple. Just 6 base pairs. So in this example, the template strand of DNA is TAC-CCC, and that codes for the messenger RNA, AUG-GGG. And those 2 codons code for the amino acids methionine and glycine. This is no different except for the fact that the enzymes used for this process are found within the host cell. In group 2, instead of double-stranded, the nucleic acid is single-stranded. So if you'll come down to the bottom, this is how it works. You have single-stranded DNA that makes a copy of itself to form a double-stranded DNA molecule. And then transcription, this is transcription here, occurs normally, and translation occurs normally. So the only difference here is that the DNA molecule in the beginning makes a complementary strand so that it will be double-stranded. Group 3 viruses. Now from group 3, well, most of the viruses from here on out will be RNA viruses. I think group 7 is a DNA virus. But group 3 viruses have double-stranded RNA. So really, there is very little difference in transcription and translation. The only difference is you can tell that this example in the beginning is RNA instead of DNA because it contains the nucleotide uracil, or U. So you've got a double-stranded RNA molecule that creates a messenger RNA molecule, that's transcription, and then the messenger RNA codes for the amino acids methionine and glycine. So in fact, since it's RNA, this is in yellow, highlighted in yellow is your template strand, and then the complementary strand is actually the same thing as what the messenger RNA codons end up looking like, just like the complementary strand to the original double-stranded RNA. All right, then we have group 4 and 5. Now, group 4 and 5 are both single-stranded RNA, but this one is great because it literally skips transcription, keeps things very simple. This is single-stranded RNA positive. So what happens here is that single-stranded RNA is the messenger RNA. So the messenger RNA is what I'm showing you right here is highlighted. AUG-GGG is already produced. There is no need for transcription. This messenger RNA carries out translation and produces the amino acids methionine and glycine. And just for a review, we would call this a dipeptide, because it's a chain of amino acids which makes it a peptide, and then it's 2 amino acids, and that's where the di comes from. So it's a dipeptide, 2 amino acids. I've made it very, very short just because I want you to understand the process, but we don't have enough time to make really long molecules like in real life. Now, group 5 viruses also are single-stranded RNA, but they are single-stranded RNA negative. So what happens there is that I've given you an example. This is the single-stranded RNA. It's actually used as the template to create a complementary strand, which will serve as the messenger RNA. So the AUG GGG, which has been our messenger RNA all along in all these examples, is produced from a single-stranded RNA. So instead of using the single-stranded RNA as messenger RNA, it copies a complementary strand, and transcription does occur. Okay, so this part is the transcription, and then translation occurs, like we've seen in all the other examples. Now, interesting. Group 6 and Group 7. These viruses are very complex. They actually contain some of their own enzymes. There's a little mistake here. I typed these up, so that little T right there, but their nucleic acid core is single-stranded RNA, but they also include reverse transcriptase, the enzyme reverse transcriptase. Well, if transcription, let's go back to the original. If transcription is when the double-stranded DNA molecule is used as a template to create the messenger RNA molecule, this part, the first part, DNA creating messenger RNA, if that's transcription, then reverse transcription would be RNA being converted into DNA. So let's see how that works in a group. I'm sorry, I had to pause, so I actually forgot what I said earlier, so I'm just going to review. In group 6, the viruses have their own enzyme reverse transcriptase and single-stranded RNA in their core, okay? So what happens is that single-stranded RNA, this is the template, copies itself into, this is reverse transcription is what this process is, into a double-stranded DNA molecule. And then that double-stranded DNA molecule now undergoes normal transcription and translation, just like in living cells. So that is group 6 viruses. HIV is a group 6 virus. And then the last one, group 7, is really strange. Here we go. Group 7 viruses actually do, just like living things in cells, they contain double-stranded DNA, but they also contain reverse transcriptase. Weird. When you already have double-stranded DNA, what do you need reverse transcriptase for? I haven't really read or learned a lot about the reasons for all these different mechanisms, but all I know is viruses have to, their goal is to replicate, is to trick a host cell into replicating them and making more and more. That's the goal of the virus. If you could say a virus has a goal, that's so viruses are more successful the more they can trick their host cell. And so more than likely these complex different ways of transcription and different types of nucleic acid cores, have something to do with that. Something to do with the host cell that they are trying to trick, whether it's a bacterium or a fungi or a plant or an animal. But anyway, this one, the double-stranded DNA is not used to make messenger RNA. The double-stranded, in fact, let's read this. The viral genome is double-stranded DNA, but the viral DNA is replicated through an RNA intermediate. The RNA may serve directly as messenger RNA or as a template to make messenger RNA. So, your examples can look very similar to, this would be without an RNA intermediate. The DNA is replicated using an RNA. And then here you've got DNA replicates into an RNA intermediate. That would be this one. And then this is your messenger RNA, which was complementary to the RNA intermediate. So you've got double-stranded DNA, RNA intermediate, messenger RNA, and then your amino acids. Really, group 7 is very, very tricky. It's just the most complicated one to understand. And but that's basically how it works. It's got double-stranded DNA and reverse transcriptase, so it doesn't carry out transcription in the same way that living cells do, because living cells don't contain reverse transcriptase. Okay, I'm going to try to go through the rest of these slides, quickly while explaining what you need to know. I went very slowly through the classifications or the groups of viruses because I think that's a concept that's kind of difficult for students to understand. So we're going to talk about viral infection. When a virus infects a host cell, it can cause what's called cytopathic effects. A pathogen is a disease-causing agent, and cyto means cell. So they are effects that can change cell functions or even destroy the cell. The symptoms result from the immune response to the virus, which attempts to control and eliminate the virus from the body. So the virus is not causing the symptoms. It's your immune system reacting to the virus that's causing the symptoms. Some infected cells, such as the cells infected by the common cold, the common cold is most often rhinovirus, die through bursting or apoptosis. Lysis or apoptosis, that's basically cell suicide. In other words, when the cell is infected with virus, the cell itself is going to be killed. It's going to be destroyed. And when the cell lyses or bursts, it releases all of the virions that are inside of it, and it can be hundreds or more. At one time, all of those virions are released, so they can go attack other cells in the same tissue. Many animal viruses, such as HIV, leave the infected cells by a process known as budding. And budding occurs, we're going to see that because that occurs in some of our living organisms. That's where the virions leave the cell individually. It's basically similar to exocytosis. A little bud forms off of the plasma membrane, and then it pinches off, and the virus is inside of it. So the cell is not immediately killed, but the cell is damaged, and it may make it impossible for the cell to function normally. And sometimes it can be used again as a host cell. This is the steps of viral infections. The steps are attachment, entry or penetration, replication, assembly, and release. And we're going to go over these steps in the next recording. But what you can see is this is a bacterial cell, and this is the bacteriophage that has attached to it. And the bacteriophage has nucleic acid in blue. And you can see that it attaches based on receptors in the virus and the plasma membrane of the bacterium. So that's how it attaches, is that the receptors match.